We are excited to share our work titled Mechanobiology in the Development and Progression of Non-Alcoholic Fatty Liver Disease, an updated review in Metabolism and Target Organ Damage. Mechanobiology is an interdisciplinary field that aims to understand how physical forces change the mechanical properties of living organisms and contribute to the regulation of biologic functions. While the development of fibrosis is an obvious mechanical change associated with the progression of non-alcoholic fatty liver disease, or NAFLD, there is evidence that disruptions of mechanical homeostasis occur in the initial stages of NAFLD, prior to the development of fibrosis. These early disruptions in mechanical homeostasis appear to significantly contribute to disease progression in NAFLD. A better understanding of the mechanobiologic alterations in early NAFLD may help us to identify new targets for the prevention and treatment of this extremely common metabolic disorder.
The liver tissue is made up of intravascular fluid, such as blood and lymph, transcellular fluid, such as bile, interstitial fluid, extracellular matrix, and liver cells, including hepatocytes, hepatic stellate cells, liver sinusoidal endothelial cells, and Kupffer cells. Liver tissue has viscoelastic properties, meaning its physical properties are determined by both its fluid and solid components. Liver sinusoids are the structural and functional units of the liver. Sinusoids are low-flow, low-pressure vascular microchannels surrounded by the cellular and extracellular matrix components of the liver. Liver sinusoidal endothelial cells form the wall of the sinusoidal vascular channels. These cells are the primary sensors of flow and pressure changes in the sinusoidal microcirculation. Liver sinusoidal endothelial cells are separated from hepatocytes by the space of Dease, which contains hepatic stellate cells, as well as interstitial fluid and extracellular matrix laid down by the hepatic stellate cells. Hepatic stellate cells wrap their long cellular projections around one or more sinusoids, and in this way contribute to the regulation of vascular tone. Kupffer cells, which are liver-specific macrophages, reside in the sinusoidal lumen to recognize molecular danger signals derived from the portal circulation. It is well known that fibrotic transformation of the extracellular matrix alters the viscoelastic properties of the liver. However, dynamic mechanical analysis has shown that pathologic accumulation of fat, meaning steatosis, as compared to no steatosis, has a profound effect on the viscoelastic properties of the liver. The increased presence of lipid droplets increases tissue viscosity. In steatohepatitis, which is a more progressive stage of NAFLD, additional mechanical changes occur, including injury to the hepatocyte cytoskeleton, resulting in hepatocyte ballooning, as well as inflammatory expansion of interstitial fluids. These changes exacerbate the volumetric squeeze of the sinusoids within the liver capsule, reducing sinusoidal space and increasing stiffness by the stress-strain response. A major consequence of these mechanical changes is increased intrahepatic vascular resistance due to external sinusoidal compression, leading to elevated portal pressure, which has been demonstrated to occur in early NAFLD, starting with steatosis and later aggravated by subsequent inflammation in steatohepatitis. Here we show in cross-section a schematized view of the sinusoidal channel in NAFLD. It is important to emphasize that, in addition to the external compression imposed upon the sinusoids by steatosis and steatohepatitis, there are a multitude of mechanical forces that result from the structural and functional perturbations within the sinusoidal microenvironment in NAFLD. These changes, such as lipid microembolization, the formation of neutrophil extracellular traps, and the formation of microthrombi within the sinusoid, as well as contraction of the hepatic stellate cells around the sinusoids, represent impediments to sinusoidal flow and contribute to increased intrahepatic vascular resistance. It has been speculated that mild elevations in sinusoidal pressure contribute to the progression of fibrosis in NAFLD, suggesting a bidirectional cause and effect relationship between portal hypertension and liver fibrosis, establishing a new paradigm in liver pathophysiology. Physical forces in the sinusoidal microenvironment influence cell function in both health and disease. All living cells have the ability to sense and generate physical forces while interacting with their microenvironment. Forces arising at the cell's interface with adjacent cells, various intravascular or transcellular fluids, and at the extracellular matrix are converted into biochemical signals and transcriptional responses through a process called Mechanotransduction. Mechanosensors, which are located primarily in the plasma membrane and nuclear membrane, detect mechanical cues within the cell itself and at its interfaces with other cells, fluids, and matrix. These mechanical cues may be transmitted to the nucleus in several ways. Mechanical cues may be transmitted through biochemical cascades involving diffusible intermediates in a process termed Mechanosignaling. These intermediates may enter the nucleus through nuclear pore complexes. The yes-associated protein, or YAP, and its paralog transcriptional coactivator with PDZ binding motif, or TAZ, have a prominent role in cellular mechanobiology in health and disease. In response to mechanical cues, YAP-TAZ translocate from the cytoplasm into the nucleus through nuclear pore complexes. In a parallel process, mechanical information propagates through the physical continuum between the contractile cytoskeleton and the nucleoskeleton. This process is termed mechanotransmission. Mechanotransmission and mechanosignaling are closely intertwined. For example, the diameter of the nuclear pore complex is modulated by stretch-mediated deformation of the nuclear envelope by the contractile cytoskeleton. The nucleus integrates all of these mechanical cues, leading to a mechanoresponse.
Mechanocrine signals are altered in many ways in NAFLD and likely contribute to all aspects of pathogenesis. Aberrant mechanoresponses may generate new mechanical forces, creating vicious cycles in the liver sinusoids. In this review, we discuss several examples of disrupted mechanical homeostasis in NAFLD that promote inflammation, angiogenesis, and fibrosis.
We hope you enjoy reading our review on the interplay between liver biomechanics and the pathobiology of NAFLD, with a focus on the early stages of disease development. We hope this review encourages further study in this area, as we strongly believe that a better understanding of this interplay may help to identify targets for the prevention and treatment of NAFLD.